The most direct path of information flow in the retina is from an input photoreceptor cell through a bipolar cell to an output retinal ganglion cell. This is also known as the direct pathway. At each synaptic connection between these cell layers, however, the neuronal responses are modulated by the lateral connections of horizontal and Amerkin cells. This is known as the lateral pathway. To understand in detail how this information processing occurs, let's first focus on the information flow from photoreceptor to bipolar cells, and then focus our understanding on retinal ganglion cell output to the optic nerve. Photoreceptors, like other neurons, release neurotransmitter when depolarized. Since the retina is part of the central nervous system, the main neurotransmitter is the amino acid glutamate. As we've just learned, photoreceptors are depolarized in the dark and hyperpolarized in the light. While this might seem counterintuitive at first, it makes sense when considering that dark rather than light is the preferred stimulus for a photoreceptor. To illustrate, when a shadow cast by an object in our surrounding passes across the retina, it responds by depolarizing and releasing neurotransmitter, thus registering the presence of the object. Since each photoreceptor cell is in synaptic contact with both bipolar cells and horizontal cells, this information can be passed on both in the direct and lateral pathways. Let's consider the transmission to bipolar cells through the direct pathway first. Based on the responses to glutamate released by photoreceptors, bipolar cells can be classified as either on or off cells. In off bipolar cells, glutamate-gated cation channels mediate a depolarizing excitatory postsynaptic potential, our old friend the EPSP, after sodium influx. In on bipolar cells, G-protein-coupled receptors respond to glutamate released by the photoreceptors by hyperpolarizing. It's worth pointing out that the labels off and on for bipolar cells refer to the fact that these cells depolarize in response to light off, more glutamate, or light on, less glutamate. Each bipolar cell receives direct synaptic input from a cluster of photoreceptors, ranging from 1 to thousands in number. In addition, each bipolar cell is also connected via horizontal cells to a ring of photoreceptors that surround the central direct cluster. Thus, the receptive field to which these cells respond consists of two parts: a circular central area providing direct input from the photoreceptors, and a surrounding area of retina providing indirect input via horizontal cells, consequently the name center surround. Generally speaking, the response of bipolar cells' membrane potential to light in the center of the receptive field is opposite to the response in the surround, irrespective of whether the cell is classified as on or off-center cell. This organization has an interesting effect of opposing physiological responses when illuminating either the center or surround with the same spot. This antagonistic surround appears to come from a complex interaction between horizontal cells, photoreceptor and bipolar cells at their synapses, and is a very active area of neuroscience research. This center-surround organization is passed on from the bipolar cells to retinal ganglion cells via synapses in the ganglion cell layer. And it's in addition modulated by the action of Amerkin cells in the lateral pathway in this layer, coordinating the integrating rod and cone inputs in the retinal ganglion cells. As mentioned previously, retinal ganglion cells are actually the only cells in the retina that fire action potentials and convey their output to the rest of the brain. There are about 1 million retinal ganglion cells in the human retina. Like bipolar cells, most retinal ganglion cells have a center-surround receptive field organization, and they receive their on or off input from the corresponding bipolar cells. Thus, an on-center retinal ganglion cell will be depolarized and fire a range of action potentials if the center of its receptive field is illuminated with a bright spot. Conversely, an off-center retinal ganglion cell will respond to a small dark spot in the center of its receptive field. Some retinal ganglion cells have more complex response profiles than simple center-surround receptive fields. For instance, some RGCs respond selectively to particular colors or movement of light patterns across local regions of the retina. This diversity of response properties derives from different connectivity patterns among the different classes of Amerkin and bipolar cells. Since there are currently over 30 specific types of Amerkin cells and about 12 types of bipolar cells known, this allows for a large combinatorial space to respond to specific features. The full parts list of different kinds of retinal ganglion cells is not yet fully known, though great progress is being made in this area today. Likewise, we don't yet know what all of these different RGC types are for. For instance, some RGCs in the retina respond selectively to different kinds of motion, but we also know that motion is processed in cortex, starting from different retinal inputs. In some cases, evidence suggests that motion-selective cells in the retina may be involved in oculomotor reflexes, such as the optokinetic reflex, which moves the eye to stabilize the image on the retina when whole-field motion is detected. In many cases, it's not yet clear how different RGCs participate in different aspects of vision, and the answer might be different in different species.